Hello everyone, my name is Prateek Sharma. Today I'm going to present on smoke-induced gut microbial dysbiosis in promoting pancreatic cancer. I have no disclosures. As we all know that smoking is a major risk factor for pancreatic cancer, but the mechanisms are still unknown. Also, smoking changes the gut microbiome, but the impact of this change is not fully explored. Our lab has recently shown the role of gut microbiome in cancer promotion. So in the similar vein, our hypothesis was that smoking promotes pancreatic cancer via gut microbial dysbiosis. And the experimental design was we took black 6 mice and gave them smoke exposure through smoking chamber for 4 weeks with or without gut microbiome depletion by antibiotic cocktail. This was followed by KPC pancreatic tumor implantation, and the tumors were followed with continued exposure. What we saw was that smoke exposure promoted pancreatic tumor growth. But what was very interesting was that the entire tumor promoting effect of smoking was gone when gut microbiome was depleted, as shown in green. The figure on the right shows that even at endpoint, there was a drastic difference in gut microbiome composition between control and smoke exposed mice. We replicated our finding with similar results using smoking analog NNK in a more rigorous mice model of pancreatic cancer, that is the KC mice model. To characterize this dysbiosis, we did stool analysis, and as shown in the figure on the left, certain bugs like Akkermansia, Clostridia, and some others were significantly enriched in smoke group. Also, the metabolome profile was very different between control and smoke-exposed mice. To evaluate if this dysbiosis on its own is sufficient to promote tumors, we did fecal matter transplant studies where we saw just FMT from tumor-naive smoke-exposed mice was able to promote tumors compared to control FMT. To evaluate the role of immune system in this dysbiosis, we did the experiment in RAG1 knockout mice which lack functional adaptive immunity. As shown in A, compared to wild-type counterparts, smoke exposure neither promoted tumor growth nor were antibiotics able to bring it down in RAG1 knockout mice. Similarly, as shown in B, smoke FMT was able to promote tumors in wild-type mice, but was unable to do so in RAG1 knockout mice, highlighting that a functional adaptive immunity is critical for this dysbiosis to promote tumors. We also did flow cytometric analysis of tumors, and as shown, that smoke exposure promoted a very immunosuppressive phenotype with decreased CD3, decreased CD8, CD4, activated dendritic cells, and increased MDSCs. However, gut microbiome depletion was able to reverse this immunosuppressive effects. Thus, in conclusion, smoke-induced dysbiosis results in an immunosuppressive tumor milieu, and gut microbiome modulation strategies, for example, can counteract this phenomenon. In terms of future direction, we would like to evaluate our hypothesis in human smokers and also find the mechanism by which this dysbiosis is promoting tumors. And thank you.